In this video, we are going to learn about blood group. Myself, Dr. NMGD, Assistant Professor, Department of Pharmacology, KLE College of Pharmacy, Hubballi. See in this slide, you can see the great scientist named Karl Landsteiner. He is from Austria. He first distinguished the blood groups in the year 1900. For this pioneer research work, he is awarded Nobel Prize in Physiology or Medicine in the year 1930. So now we are going to discuss about the blood groups. What are the importance of blood transfusion? All we know about the normal blood volume in healthy adult. See, that is approximately 5 liters. See, this volume of the blood, it is always maintained by homeostasis mechanism. When there is a decreases in the volume of the blood, that may leads to number of consequences like decreases in the blood pressure, low perfusion, less oxygen supply to the organs, that may lead to the hypoxia and other organ failure. So in order to maintain the blood volume, so for example, during the accidents or burn or because of illnesses, when there is a decreases in the blood volume, in order to maintain the blood volume, we have to transfuse the blood from the donor to the patient or recipient. See, if the blood is not compatible, see, that may leads to blood transfusion reaction. See, that may be fatal to the recipient. Not all blood groups are compatible with each other. When the receiver of a blood transfusion has antibodies against the donor blood cells, that causes blood clumping, or that is also called as agglutination. That is due to the immunological reaction. So this process, it is called as a blood transfusion reaction. The clumped red cells can crack and causes toxic reactions, which is dangerous for individual or the recipient. Then what are the different blood groups? On what basis the blood is divided into different groups? The differences in human blood are due to the presence or absence of certain surface proteins. They are called as the antigens. And also some of the antibodies which are produced by the immune system against the antigens. Antigens are located on the surface of red blood corpuscles, and the antibody is normally present in the blood plasma. See, that is released by the immune system. The blood group is inherited from your parents. ABO blood grouping system. According to the ABO blood group system, there are 4 types of blood groups. Blood group A, B, blood group AB and O. Again in the A positive, A negative, B positive, B negative, AB positive and AB negative, and O positive as well as the O negative. So these are the different types of blood groups. So on what basis the blood is divided into these groups? See, that is mainly based upon the antigens, mainly the A antigen and B antigen, and also the one more antigen, that is the Rh antigen. See, these antigens, if they are normally they are present on cell membrane of red blood corpuscles. See, in this figure, you can see the different blood groups A, B, AB and O blood group. Okay. See, the immune system that may produce the antibodies. So normally this type of antigens which are present against the same antigens, the antibodies normally not produced by the immune system. Let us see the one by one. The blood group A contains A antigens on the RBCs, and immune system of this blood able to produce the antibodies B, not the antibody A. Then blood group B. Here you can see the blood group B contains B antigens on the red blood corpuscles, and immune system of this blood able to produce A antibodies, not B antibodies. The next blood group AB. Here you can see both the A and B antigens which are present on red blood corpuscles, but immune system of the AB blood group, they are not producing anti-A antibodies or anti-B antibodies. The next is blood group O. In this blood group O, see, there is no A or B antigens on red blood corpuscles, but immune system of this blood is able to produce A as well as B antibodies. See, these are the 4 blood groups on the basis of antigen A as well as the B and antibodies A and antibodies B. And Rh group, normally the A positive or A negative, B positive or B negative, AB positive or AB negative, O positive and O negative. Plus or negative mark we are mentioning in front of A, B or AB or maybe the one O. Here what happens, the one more type of antigen, if that is present on the red blood corpuscles, this antigen it is called as Rh or D antigen, or that is also called as agglutinogen. So because of first time this type of antigens they are identified in the rhesus monkey in the year 1940. If this type of antigen is present on red blood corpuscles, that blood group is the Rh positive. If this antigen is absent, that blood group is negative. Here you can see the importance of Rh antigen. A person with Rh positive blood, he or she can receive Rh negative blood without any problem. Means he can receive both Rh positive as well as Rh negative without any problem. But what happens when a person with Rh negative blood, he or she only can receive Rh negative blood group. If he or she receive Rh positive, the antibodies which are produced against Rh antigen, that may lead to agglutination. clumping of blood that is occurs. So now we are going to discuss about how we are going to identify different blood groups. That is A positive, A negative, and so on and so forth. Here you can take the glass slide with 3 grooves. Add 1 drop of A antibodies in the first and B antibodies in the second and RH antibodies in the D or in the last groove. Then after taking 1 drop of antibody A, B and RH, add 1 drop of blood here and 1 drop of blood in the B and 1 drop of blood in the D. Take another glass slide. By using the different edges of another glass slide, just mix up the blood and the anti-sera or the antibodies. See here the clumping, if the blood is compatible, means if there is no clumping or agglutination in all 3, see that is O negative blood group. If the clumping, if there is no clumping here, no clumping here, means that blood group is O. If there is no clumping here, means that is RH negative. Okay. See, if only clumping here means that blood group is A, only clumping here means that blood group is B. Clumping in both these two, that is blood group AB. See, if there is no clumping in both these two, that is a blood group O. See, clumping here, that is RH positive. If there is no clumping here, means that is RH negative. See, this is the way how we are going to identify the different blood groups. So one more important point with respect to transfusion of blood, that is compatibility. Before going to transfuse the blood to the recipient, so we are checking the compatibility of the recipient as well as the donor's blood. So in order to see the compatibility, see other than these A and B and RH antigens, if there are other antigens which are present in the blood of a donor, sometimes that may also leads to agglutination. Just by mixing the drop of blood of donor as well as the recipient, and we are observing the compatibility. If the blood is compatible with the recipient, then only the blood transfusion, so that is done. See in this slide, you can see the different blood groups, and so these blood group persons can receive from these blood groups of donors. Here you can see one by one, the O positive blood group person, he can receive from O positive and O negative. O negative individual can receive blood from only from O negative. A positive person receive blood from A positive, A negative, O positive and also O negative. And B positive individual can receive blood from B positive, B negative, O positive and O negative. And similarly from B negative, he can receive from these blood groups. AB positive receive blood from these groups, and AB negative can receive from these blood groups. So these are the references. Thank you.